Hi, I'm Brynn FitzGerald.
And I'm Henry Pohlman. And so we are here to give about a 15-minute presentation. We were interested in the neurobiology of disease, and specifically Alzheimer's disease. We figured that's a pretty relatable thing. A lot of people are affected by it, a lot of parents, grandparents. So moving into our specific study is area-specific alterations of synaptic plasticity in the 5X FAD mouse model of Alzheimer's disease. Dissociation between the somatosensory cortex and the hippocampus. So moving right into it. So hopefully everyone here has had some introduction to Alzheimer's disease. You've at least heard of it. But just to get us all on the same page, we'll do a quick background. So Alzheimer's disease is a progressive neurodegenerative disease. It's actually one of the most common forms of brain degeneration, commonly linked to aging. And so on a cellular level, there are three primary components that are generally associated with Alzheimer's disease. So the first, which is the most traditional form of Alzheimer's, is these AB-senile plaques, which stands for amyloid beta peptides. The second thing are these neurofibrillary tangles. And then finally is the hyperphosphorylation of tau proteins. Tau proteins, you can kind of think of them as like they're like a scaffolding protein in the cell. But for this paper, we're going to be focusing on these amyloid beta peptides, these plaques that are formed by them. Cognitively, Alzheimer's is generally linked to this deficit in learning and memory. Very specifically, it's generally linked to this deficit of long-term potentiation, which is the formation of long-term memories from short-term memories. So generally, people with Alzheimer's are not able to form long-term memories. And of course, if we're dealing with a disease that deals with this dissociation of memory and learning, generally the areas of the brain that are going to be affected are going to be dealing with learning and memory, which is, of course, the case. So two of the areas that are most affected by Alzheimer's are the hippocampus, which is in previous research been shown to be very, very critical in the formation of memories. And then also the cortex, which specifically in this study is looking at the somatosensory cortex. The somatosensory cortex is the front of the parietal lobe. There is the primary and secondary. The primary is more anterior to the secondary. So right in this region, here's the front of the brain. So right here at the front of the parietal lobe. So moving now specifically into this study, so we were looking at these amyloid beta peptides, or these A-beta peptides, AB peptides. So these are the primary component of these amyloid plaques that form. And these plaques actually form extracellularly and intracellularly. And the overproduction of these plaques is one of these more traditional primary links of the cause of Alzheimer's. And specifically in this study, so we're going to be looking at this loss of synaptic plasticity in these two specific brain regions, the hippocampus and the somatosensory cortex, as they link to the overproduction of amyloid beta peptide. Now a little bit about the mice that were used. So these 5X FAD mice. The reason that they're called 5X FAD mice is because they have 5 mutations, these familiar Alzheimer's disease mutations, 5X FAD familiar Alzheimer's disease. Of these, 3 of the mutations come from humans actually, they're human mutations, 2 of them from mice. Interestingly enough, these are some of the best models to use for studying Alzheimer's because they develop Alzheimer's very, very quickly. So within 2 months of birth of these mice, they're already showing signs of neural degeneration within these specific areas, the hippocampus and the somatosensory cortex. Then at 6 months, we do see full brain degeneration, which I would link to like a photo like this. Hopefully some of you guys may have seen this. So this is kind of a classic image of Alzheimer's, this total brain degeneration. So that would be occurring at 6 to 12 months. But even at 2 months, we are seeing the beginning of brain degeneration linked to Alzheimer's in these specific areas of learning and memory. And then finally, just a little bit, so this general purpose of this study is essentially, within these 2 different areas of the brain, there are specific types of these neural plaques that are being formed, and they're different from one another. And so the researchers wanted to look at, is there a correlation between the amount of synaptic plasticity that can occur and their correlation to these different types of neural plaques that are occurring in these different areas. Interestingly enough, these researchers also developed they founded a new technique, which is called this multi-electrode array. It seems pretty simple. It's actually interesting that they were the first ones to develop this. Essentially, they took a sagittal brain section of both the layer 5 of the somatosensory cortex, which is what they studied, and area CA1 of the hippocampus. And essentially, by taking a sagittal brain section, they were able to, in one brain slice, take areas of both of these sections. So they were able to study them simultaneously. Seems simple, but they were very specific to point out that they were the developers of this new technique.
Alright, so real quick I'm going to go through the methods of this paper. So as Henry mentioned, they used transgenic mice with these 5 different mutations. that are related to Alzheimer's disease. And so they used 6-month-old male mice, and so to compare this mouse model of Alzheimer's, they used litter mates from these mice that were wild type, so they didn't have any mutations as controls. So they took sagittal brain slices, and these brain slices went through a variety of immunostaining, imaging, and the experiments were actually done on the brain slices. And so from each of these slices, they recorded the number of plaques per square millimeter in the cortex and the hippocampus. And along with recording the number of plaques, they also differentiated between diffuse plaques and dense core plaques. So you see the image on the right. You see the diffuse plaque, it's very spread out, and the dense core is very, very tight-knit, very dense. And we'll go into more of that later. And so they took images of these cortico-hippocampal slices to map the actual synaptic signals. And so using this multi-electrode array, they stimulated the collateral pathway of the hippocampus and also the cortex using these electrodes. They recorded field EPSPs and population spikes, and they compared the slopes of these field EPSPs that they recorded. And so they were looking at a couple different things. They looked at baseline synaptic signals, which they encouraged using 0.066 hertz frequency stimulation from the electrodes. They looked at short-term plasticity, and so to do that, they did 2 stimulations with an interpulse duration of 25 milliseconds to 500 milliseconds. And then their big thing was long-term potentiation, which we'll go into more later also, but they used 1 or 3 theta burst stimulations with a 30-second interval in between. Just something to note, for some of their recordings, they used picotoxin, which was a GABA-A receptor blocker. And then statistically, they used either one-way or two-way ANOVAs to compare different findings.
Okay. So moving to the results, so just to give you guys a little bit of a roadmap, there are 3 large sections of the results. We're first going to look at the morphology of these different plaque types and their association to the different brain regions. The second thing we're going to look at is the basal transmission and short-term plasticity, so short-term memory and then just a basal synaptic plasticity line. And then finally, the meat of our findings is going to be this long-term potentiation, because that is really what's important in Alzheimer's, is this inability to produce long-term memories. So moving right into it, so this first section of results looks at the morphology of these different plaques. And so the first thing I want you to look at is figure 1E. And so what figure 1E shows is plaques per millimeter squared. So essentially how many plaques are in each of these brain regions. And we see no significance between them. Both of these are the same. So essentially in both the cortex, the somatosensory cortex, and the hippocampus, there are the same amount of plaques. The difference occurs in their morphology. So what we did find is figure 1F shows us this ratio of diffuse to dense plaques. And what we find here is we find a very significant difference in that the cortex is full of these diffuse plaques. So every plaque actually has a dense core. The only difference is that what's considered a diffuse plaque has this diffuse corona of more basically plaque material surrounding the dense core. So what we find is that in the cortex we have not only a dense core but also this diffuse corona around it, while as in the hippocampus we only have the dense core. So that's the primary difference of the morphology of these plaques between the 2 brain regions. Moving into our second part of the results. So now we're going to look at basal transmission and short-term plasticity. The first thing I want you to note is when we look at basal transmission, we're going to be looking at FEPSPs, which is functional excitatory postsynaptic potentiation. So basically, how strongly is the connection going through these synapses? And what we find here is what we should be finding, is that the colored in dots are the wild type, whereas the hollow dots are the 5X FAD mice, so our experimental mice. And in both the hippocampus and the cortex, we find that the wild type scored significantly better than the 5X FAD mice. This seems like a reasonably trivial finding, but it's important to have because it basically shows that, yes, our 5X FAD mice do have a negative, these mutations do have a negative effect in the ability of forming synaptic potentiations. Now we're going to move into short-term plasticity. Interestingly enough, short-term plasticity, well, it did have interesting curves, as we can see here. At no point were they significantly different from each other in the 5X FAD or the wild type. And so what this shows is that actually these 5X FAD mice, which did have Alzheimer's, which was shown here because of the loss of synaptic potentiation in terms of basal transmission, but it did not affect their short-term memory, essentially. Interestingly enough, there are some interesting curves in that in the hippocampus the ratio goes up, at about the at about the 50 interpulse duration, whereas in the cortex it actually dips down. I found that interesting, so did the researchers. They didn't really touch on it, possibly an area for future studies. So yeah, that's essentially that, so that we found no difference for the short term. So now moving into the meat of our findings, which is long-term potentiation. So the first thing is that picrotoxin was used. So picrotoxin is a GABA, is a GABA inhibitor. GABA is the primary inhibitory neurotransmitter. You inhibit an inhibitor, it's going to basically, it's going to be an excitatory. It's good for causing some synaptic transmission to occur. So the first thing I want you to note is that we did see some activity in the 5X FAD, but we did see significantly more activity in the ability to form long-term potentiation in the wild type. This is, of course, as expected. And in the cortex, we saw no potentiation in either, so there was no change. This is the 1 theta burst stimulation with picrotoxin. 1 theta burst stimulation without picrotoxin, the data was not shown, had nothing. So in both the hippocampus and the cortex, there was no significance. It basically mimicked just the cortex for this. Moving now into the next slide. So now we're gonna keep pumping up the intensity of our stimuli. So now we're going to look at 3 theta burst stimuli without picrotoxin. So of course, mimicking the findings of 1 theta burst with picrotoxin, we did see that the wild type mice did score significantly better than the 5X FAD. Once again, the 5X FAD showed no activity. Again, in the cortex, we saw no activity in either. So essentially, we're still only in the hippocampus is where long-term potentiation is occurring. So basically, we got to keep ramping up the stimulation. So now let's do 3 theta bursts with picrotoxin. Last slide. So now finally, we're going to see some activity in these 5X FAD mice. So of course, as you would expect, the wild type did score significantly better. This is on par with all the other findings we've had. Interestingly enough, finally, we do see some long-term potentiation occurring in the 5X FAD mice. You can see here some dash marks, which does indicate that there was significance, and ANOVA was done against the other 2, the 1 theta burst with picrotoxin and the 3 theta burst without, and we did find that it was significantly higher. So that's interesting. So we're actually starting to get long-term potentiation occurring in the hippocampus. Interestingly enough, again, finally, we are going to start to see some activity in the cortex, although this is only with the wild type. So once again, the wild type did score significantly better, and we never really saw any activity in the cortex with the 5X FAD mice, which we'll discuss in the discussion a little bit more.
All right, so real quick, going through the discussion, the main points of the paper. As we talked about earlier, the reason they start the discussion talking about why, again, they use these transgenic mice, and it was because they have an accelerated timeline for the symptoms of Alzheimer's compared to other mouse models and other models of Alzheimer's that researchers have been using. So overall, they were looking at different electrophysiological characteristics between these 5X FAD transgenic mice compared to the wild-type control, specifically in the CA1 region of the hippocampus and the somatosensory cortex, or layer 5 of the somatosensory cortex. And so just looking at basal synaptic transmission, they found that in the hippocampus and the somatosensory cortex, they found changes in the basal AMPA receptor-mediated synaptic transmission in these 5X FAD mice. When they switched and started looking at short-term plasticity in the hippocampus and the cortex, they found no difference. And so if there had been a difference in short-term plasticity, there's 2 ways that could have been shown, through facilitation or depression. And facilitation would have been shown looking at the CA1 region of the hippocampus, and that would have come from basically residual elevation of calcium ion levels. It also could have been depression, and that would have been seen in the cortex, and that would have come from basically depletion of readily available vesicles. And so since facilitation and depression both would have had to do with presynaptic sites, and there was no change in short-term plasticity, the researchers suggest that, at least in this model of Alzheimer's, presynaptic sites aren't affected. And so their big finding was with long-term potentiation, or LTP, and they found that in this mouse model of Alzheimer's, in the hippocampus, LTP was drastically reduced, but in the somatosensory cortex, LTP was actually completely gone. And so they conclude that cortical plasticity is much more damaged than hippocampal plasticity in these 5X FAD mice. They end their discussion looking at a couple reasons why LTP might have been impaired. First, they start, I have a quote up here, there is an inverse correlation between the occurrence of long-term synaptic plasticity and the proportion of A-beta depositions with diffuse corona in these areas. So the higher proportion of A-beta depositions that had a diffuse corona, the worse off long-term synaptic plasticity was. And so they give a couple different reasons why that might be true. They talk about how in the diffuse corona you have these distended neurites, indicators of neuronal injury, which might be affecting the synapses. You have synaptic vesicles, which might be causing a disorganization of different connections. And they also talk about how A-beta depositions actually impact AMPA and NMDA receptors. And so for the AMPA receptors, A-beta depositions actually encourage their endocytosis. With the NMDA receptors, A-beta has a direct agonistic effect. And then finally, they talk about how, just in general, cortical networks might be reorganized because of these depositions, mostly in terms of inhibitory interneurons are affected the most. And so overall, this paper created a useful model of Alzheimer's, and also looking at their general conclusion that cortical plasticity is affected more, that correlates well with a lot of the impairments of cortex-dependent behaviors we see occurring with Alzheimer's disease. And so, as we mentioned earlier, they have this new method, this multi-electrode array, as a way of recording simultaneously from different brain regions. And so using this method, not only for Alzheimer's, but also for other neuropathologies, we might be able to create a better understanding of the molecular components of these pathologies, but also create a more targeted approach to treating them.
Thank you.
Thank you.